So my name is Nerea, and I'm here to talk about alternative proteins. And in case you're wondering, no, Donald Trump did not discover them, although he does like some alternative facts. So to talk about alternative proteins, we should first understand what a protein is. And for those of you that you don't know much about molecular biology or genetics, there's this rule in the molecular biology field that says one gene can only make one protein, and only one. So to understand how proteins are made, I'm gonna take some volunteers, but you don't need to move, so with this big table. So let's imagine you all guys are letters from the genome code, so A, T, C, G, and that's it. You're only 4 letters. So let's imagine you're a gene. You're the first one, and I'm a big fatty ribosome. And I come here and I see a start sign, so I say, yeah, I can start producing proteins. And because I'm a ribosome, I like threesomes. So I'm gonna pair you in 3s, and I'm just gonna choose you 3 first. This way. So you lucky 1, and you 2. So you 3 are going to be an amino acid, and I'm going to say first amino acid, and then I'm going to go to the next 3, so the next 3 Ys, and I'll put another amino acid, and so on until I find a stop sign that says, that's it, there's no more protein. So that's it, that's the rule in science. One gene makes one protein. Now let's say there is another start sign that I don't see, and it's like behind this guy. So another fatty ribosome comes here and then says, oh, there is a start sign, so I'm gonna start making threesomes. Now, the threesome that I make here is not the same one that the first ribosome made, which was this one. So with the same code, another ribosome can make another protein because the threesomes are different. So there's variety, international and stuff, all very nice. So as we see, although the rule for us, the researchers, is just one gene, one protein, we have just proven now that you can make different combinations. So that is alternative proteins. That is proteins that don't follow the rule of the molecular biology. So now, why are these important? It's because when researchers discover a gene, they annotate it on a big database online, and they assign a protein. And researchers that do research on cancer or diabetes, neurodegenerative diseases, they go to that database and say like, oh, I'm gonna do research on this protein. But they are ignoring the other alternative proteins that can be potential to discover cool things in cancer or diabetes. So there is a big gap in our knowledge that we ourselves are making by ignoring and excluding these alternative proteins. For example, when I was doing research back in Quebec, I was doing research in ALS, which is the disease that Stephen Hawking had. And we were doing research on an alternative protein that could be potential to for the treatment of this disease. And my job was to study 4 mutations that had been ignored because in the genetic code, they were not relevant for the normal protein, but they made a big change for the alternative protein. So they could have a potential impact on the cure of this disease. And what this teaches us is that we just proving here is that we might need to change the big rule in science and biology and say that one gene can make a lot of proteins because we can have different combinations. And also it proves us that sometimes alternative facts are worth to look at. So thank you very much.